Good day, everyone. Today we're going to discuss the factors that inhibit or favor the growth and survival of microorganisms in food. I am Angelica Cabantag, and with me are Miss Kaba, Miss Buhayan, and Miss Kabugnason. The interaction between microorganisms and other living things is natural, constant, and which plays a significant role in maintaining the ecological balance and stability of biogeochemical cycling. Take, for example, food. Food acts as good medium for transmission of many diseases and infections. The growth of microorganisms in food is dependent on various parameters. The factors influencing their growth are physical, chemical, and biological in nature. So today we're going to discuss the intrinsic and extrinsic factors that influence their growth. Depending on the type of microorganisms, the growth of many organisms in food can result in improving overall quality of food, and in some cases, they can deteriorate the quality also. Take, for example, the ERGOT blight during the SALEM witch trial in the late 1600s. The ERGOT is a fungi that causes hallucination and causes the person infected to behave rather oddly. Hence, those infected are accused as witches. But not all microbes present in food are bad guys. Take, for example, the creation of cereal. So, one night, William Kellogg accidentally left a dough on the table. With enough moisture and optimum temperature, some molds grew in the dough the next morning, which then leads to the creation of cereal. So with these examples, you already know the importance of these intrinsic and intrinsic factor which influence the growth of microorganisms. So let's discuss intrinsic parameters. These are parameters present in substrates or food in which microorganisms are growing. First is the pH, or if you want to sound intellectual, the hydrogen ion concentration. So all organisms have a minimal, maximal, and optimal pH for their growth, survival, and activity of their enzymes. Growth of microorganisms is affected by the pH of the growth medium, resulting to large number of enzymes responsible for metabolism and growth. Influence of pH of food not only has effect on growth of microorganisms, but also on processing conditions. Food having acidic contents promotes growth of acid-loving microorganisms such as yeast, molds, and some acidophilic bacteria. So as you can see in the picture, a food with an acid pH would tend to be more microbiologically stable than neutral or alkaline food. Because of this restrictive pH, the foods such as soft drinks or sauerkraut and pickles are stable against bacterial spoilage. So, as you know, those fermented food are not really susceptible to spoilage. And most of the bacteria, except acid fermenters, favored alkaline or neutral pH.
The next one is water activity or moisture content. Water is an excellent solvent for all life processes. The amount of water required varies for different organisms, and this is expressed as available water or water activity, or A subscript W. Water activity is the vapor pressure of the solution divided by the vapor pressure of the solvent, expressed as AW is equal to P over P naught. So as you can see, pure water has an AW of 1.0, and the water activity of most fresh food is 0.99, and in general, bacteria requires more water activity than molds and yeast. And most of the food's spoilage bacteria do not grow below 0.91, while spoilage molds can grow even at 0.80. So you must remember this, that gram-negative bacteria have higher water requirements than gram-positive bacteria. So as you can see, a Staphylococcus aureus, which is gram-positive, has a water activity of 0.86, while Clostridium botulinum, which is gram-negative, has a very high water activity, which is 0.94. So you should all remember this. The next intrinsic factor is the redox potential. The reducing and oxidizing power of the food will influence the type of organism and chemical changes produced in the food. So there are four characters that determine the redox potential of food. First, oxygen tension of atmosphere above the food, access of atmosphere to the food, resistance of food to the changes occurring, and the OR state of materials present in food. But for today, we're going to talk about the OR. So the OR potential is written as EH. If the substrate is highly oxidized, it would have a positive EH. And if the substrate is reduced, it will have a negative EH. So on the table, you can see the aerobic and anaerobic bacteria that grow on the different OR potential. So on the left, we have the aerobic bacteria, bacilli, cocci, micrococci, pseudomonas that grow on positive OR potential. And on the right, we have anaerobic bacteria such as Clostridium and Bacteroides that grow on negative OR potential. So you must remember that fresh vegetables, fruits, and meat promote the growth of aerobic microorganisms in the surface regions because of the positive redox potential. Again, they grow on the surface region because of the positive redox potential. And on the other hand, anaerobic microorganisms grow in the inner parts of the vegetables, fruits, and meat because of the negative redox potential.
Microorganisms require certain basic nutrients for growth and maintenance of metabolic functions. The amount and type of nutrients required range widely depending on the microorganism. Nutrients are one of the most important compounds for the growth and functioning of microorganism. Nutritional quality of food depends on the chemical composition, nutritive value or nutrients, their proportion and growth promoting ability to the microorganisms. So in this part, we are going to talk about the composition of nutrients. The most important factors which have to be considered are the energy substances in food, nitrogen substances, growth promoting substances, accessory food substances or vitamins, minerals, and water content, which are all very essential for growth or energy production of microorganisms. The most energy sources of organisms are carbohydrates. Complex carbohydrates such as cellulose, hemicellulose, starch, pectin can be utilized by various types of microorganisms. At the same time, carbon compounds such as esters, alcohols, peptides, amino acids, organic acids, and their salts serves as energy source of microorganisms. Bacteria are identified and classified based on their ability or inability to utilize various sugars and alcohols. Most organisms can hydrolyze complex carbohydrates and can use glucose as an energy source. And some organisms have the ability to hydrolyze pectin by producing the enzyme pectinase. Hydrolytic products of proteins and peptides serves as sources of nitrogen for many proteolytic bacteria such as Pseudomonas. Some microorganisms can hydrolyze triglycerides and other type of fats by microbial lipase and produces glycerol and smaller fatty acid. In this step, triglycerides are hydrolyzed into diglyceride, then monoglycerides and glycerols under alkaline condition by microbial lipase. The glycerol and fatty acids are excellent sources of carbon and energy sources of many aerobic organisms. Hydrolytic products of proteins and peptides serves as sources of nitrogen for many proteolytic bacteria such as Pseudomonas. Amino acids serve as a source of nitrogen and energy and are utilized by most microorganisms. Some microorganisms are able to metabolize peptides and more complex proteins. Other sources of nitrogen include urea, ammonia, creatinine, and methylamines. Some microorganisms require vitamins and other growth factors for their growth, and that has to be supplied with their growth medium. Such microorganisms are called fastidious organisms, and their food contains different vitamins, minerals, and other growth factors, and their composition and content may vary. Fresh plant and animal food contain vitamin B complex and fruits are low, but fruits are high in ascorbic acid. Processing of food often reduces the vitamin content. Thiamine, pantothenic acid, folic acid, and ascorbic acid are heat labile, and drying causes the loss of vitamins such as thiamine and ascorbic acid. Storage of food for long may also result in a decrease in vitamins and other growth factors. Some microorganisms produce vitamins and other growth factors which support growth of other organisms present in food, as each kind of microorganisms has a range of food requirements. Water is another very important component for food nutrients. The water requirement of organisms will depend on the type of microorganisms. Generally, molds have the lowest requirement, followed by the gram-negative bacteria, yeast, and then the gram-positive bacteria. Examples of minerals that is required for microbial growth include the phosphorus, iron, magnesium, sulfur, manganese, calcium, and potassium. In general, small amounts of these minerals are required, thus a wide range of foods can serve as good sources of minerals. The microorganisms that usually predominate in foods are those that can most easily utilize the nutrients present. Generally, the simple carbohydrates and amino acids are utilized first, followed by the more complex forms of these nutrients. The complexity of food in general is such that several microorganisms can be growing in a food at the same time. The rate of growth is limited by the availability of essential nutrients. The abundance of nutrients in most foods is sufficient to support the growth of a wide range of foodborne pathogens. Some foods intrinsically contain naturally occurring antimicrobial compounds that convey some level of microbiological stability to them. There are a number of plant-based antimicrobial constituents, including many essential oils, tannins, glycosides, and resins that can be found in certain foods. Specific examples include eugenol in cloves, allicin in garlic, cinnamic aldehyde and eugenol in cinnamon, allyl isothiocyanate in mustard, eugenol and thymol in sage, and carvacrol and thymol in oregano. Other plant-derived antimicrobial constituents include the phytoalexins and the lectins. Some animal-based foods also contain antimicrobial constituents. Examples include lactoferrin, conglutinin, and the lactoperoxidase system in cow's milk, lysosome in eggs and milk, and other factors in fresh meat, poultry, and seafood. So lysozyme is a small protein that can hydrolyze the cell wall of bacteria. The lactoperoxidase system in bovine milk consists of three distinct components that are required for its antimicrobial action. First is the lactoperoxidase, then the thiocyanate, and the third one is the hydrogen peroxide. Gram psychotrophs such as the pseudomonads have been shown to be very sensitive to the lactoperoxidase system. Plant and animal-derived foods, especially in the raw state, have biological structures that may prevent the entry and growth of pathogenic microorganisms. Examples of such physical barriers include the testa of seeds, skin of fruits and vegetables, shell of nuts, and egg cuticle, shell, and membranes. Plant and animal foods may have pathogenic microorganisms attached to the surface or trapped within surface folds or crevices. Intact biological structures can be very important in preventing entry and subsequent growth of microorganisms, and several of these factors may influence the penetration of these barriers. Fruits are an example of the potential of pathogenic microorganisms to penetrate intact barriers. At harvest, pathogens will survive but usually not grow on the outer surface of fresh fruits and vegetables. Growth on intact surfaces is not common because foodborne pathogens do not produce the enzymes necessary to break down the protective outer barriers on most produce. These outer barriers restrict the availability of nutrients and moisture. The egg is another good example of an effective biological structure that, when intact, will prevent the external microbial contamination of the perishable yolk. Contamination is possible, however, through transovarian infection. For the interior of an egg to become contaminated by microorganisms in the surface, there must be a penetration of the shell and its membranes. In addition, the egg white contains antimicrobial factors. So when there are cracks through the inner membrane of the egg, microorganisms penetrate into the egg. And factors such as the temperature of storage, relative humidity, age of eggs, and level of surface contamination will influence the internalization. Heating of food as well as other types of processing will break down protective biological structures and alter such factors such as the pH. And these changes could potentially allow the growth of microbial pathogens.
Good day, everyone. I am Shiname M. Kabugnason, and let's proceed to extrinsic factors. The extrinsic parameters of foods are those properties of storage environment that affect both foods and their microorganisms. In other words, they are those that refer to the environment surrounding the food. So the first extrinsic factor is the relative humidity. Relative humidity is the amount of moisture in the atmosphere or food environment. When foods with low water activity contents are placed in high relative humidity environments, the food take up more moisture until equilibrium has been established. It means that their water activity increases and the food gets spoiled easily. Like for example, dry grain stored in an environment with high humidity will take up water and undergo mold spoilage. And foods that undergo surface spoilage from molds, yeast, and certain bacteria should be stored under conditions of low relative humidity. So the next factor is the atmospheric gases. Gases inhibit microorganisms by two mechanisms. First, they can have a direct toxic effect that can inhibit growth and proliferation. And carbon dioxide, ozone, and oxygen are gases that are directly toxic to certain microorganisms. This mechanism is dependent upon the chemical and physical properties of the gas and its interaction with aqueous and lipid phases of the food. For instance, oxidizing radicals generated by ozone and oxygen are highly toxic to anaerobic bacteria and can have inhibitory effect on aerobes depending on their concentration. Carbon dioxide can be used to control microorganisms in food, and ozone has high antimicrobial properties, and it has strong oxidizing property, hence it should not be used for fat-rich food as it will undergo auto-oxidation. The second inhibitory mechanism is achieved by modifying the gas composition, which has indirect inhibitory effects by altering the ecology of the microbial environment. When the atmosphere is altered, the competitive environment is also altered. Atmosphere that have a negative effect on the growth of one particular microorganism may promote the growth of another. Nitrogen replacement of oxygen is an example of this indirect antimicrobial activity, and studies shows that controlled atmosphere and modified atmosphere packaging of certain foods can dramatically extend their shelf life, and modified atmosphere package or MAP technology is commonly used for this study.
Good day everyone, I am Chanmal Sibuhayan and I will be discussing about the third and fourth extrinsic factor that inhibit and favor the growth of microorganisms in food. Microorganisms can grow over a wide range of temperatures. The lowest temperature at which a microorganism has been reported to grow is negative 34 degrees Celsius, and the highest is somewhere in excess of 100 degrees Celsius. But some spore-producing bacteria such as Bacillus, Staurothemophilus, Clostridium tetani, and Clostridium perfringens can grow above 100 degrees Celsius. Based on the temperature range, microorganisms are classified as three groups: psychrophiles, mesophiles, and thermophiles. Psychrophiles are those organisms are grown between the temperature ranges of 2 degrees Celsius to 20 to 30 degrees Celsius. The psychotrophs found most commonly in foods are those that belong to the genera Pseudomonas and Enterococcus. Mesophiles are organisms preferably grow at a temperature between 20 degrees Celsius and 45 degrees Celsius. Mesophilic species and strains are known bacteria among all genera and may be found in food held at refrigerator temperatures. The thermophiles are organisms that grow better in range of 55 degrees Celsius to 65 degrees Celsius. Most commonly, psychotrophs are Alkaligenes, Shawonella, Brocothrix, Corynebacterium, Flavobacterium, Lactobacillus, Micrococcus, Pectobacterium, Pseudomonas, Psychrobacter, Enterococcus, and others. We also have mesophiles organisms, the Listeria monocytogenes, Staphylococcus aureus, and Escherichia coli. Other examples of species of mesophiles are Clostridium cloyveri, Pseudomonas maltophilia, Theobacillus novellus, Streptococcus pyogenes, and Streptococcus pneumoniae. Thermophilic bacteria are most commonly in genera Bacillus, Penibacillus, Clostridium, Geobacillus, Cyclobacillus, and Therminoaerobacter. The fourth extrinsic factor is the other microbial flora. Microorganisms present in the food can undergo various types of negative interactions. These kinds of interaction cause inhibition of some microorganisms as they are undergoing competitions and antibiotics. Some organisms, especially molds, can produce various types of secondary metabolites such as antibiotics that are toxic to many bacteria. Some foodborne organisms produce substances that are either inhibitory or lethal to others. These include bacterioxins, hydrogen peroxide, and other organic acids.